The next assessment statement says, state that photosynthesis consists of light-dependent and light-independent reactions. So these are the two, and we'll explain these in more detail. But for now, you have to know that there are two sets of reactions. The one on the left is the light-dependent reactions. It requires light, and that occurs in the thylakoid membrane. And on the right, the light-independent reactions, also known as the Calvin cycle. This occurs in the stroma of the chloroplast. So let's look more specifically at the light-dependent reactions. You have two photosystems, photosystem II and photosystem I. You see here that when light hits the chlorophyll that's on the stroma side of the thylakoid membrane, it excites the electrons. And you see this blue dotted line is the pathway of the electrons through this electron transport chain. When the electrons get pulled by these other factors in the electron transport chain, chlorophyll loses the electrons, and that has to get replaced by something. It gets replaced by parts of the electron transport chain that like to donate electrons. And eventually, those electrons come from water. Water is split into oxygen and hydrogen ions, and those electrons flow through this electron transport chain. The molecules that are after the initial ones like to accept them more than the ones before that in the electron transport chain. And then when it gets to this cytochrome B6F complex, it pumps hydrogen ions from the stroma into the thylakoid lumen, concentrating the hydrogen ions in the thylakoid lumen. The electrons keep flowing through this electron transport chain because factors like this plastocyanide, which you don't need to know, are accepting the electrons. They want the electrons more than the ones before it. The electrons get to the photosystem 1, indicated here by PS1, and the electrons have to get excited again. They're excited again by the light hitting the chlorophyll that's sitting on the stromal side of the thylakoid membrane. It excites the electrons to that point, and the ferredoxin, which you don't need to know, accepts the electrons, and it wants the electrons more than the chlorophyll does. Eventually, these electrons are accepted by NADP+, which becomes NADPH. It accepts the electrons most readily. When the hydrogen ions are accumulating in the thylakoid lumen, they flow through an ATP synthase transmembrane protein, and that flow of hydrogen ions back into the stroma, it is used by this ATP synthase molecule to take ADP and make it ATP. And both the NADPH and the ATP are used in the Calvin cycle, the light-independent reactions. State that light is used to produce ATP and to split water molecules, which is called photolysis, right? Photo means light, lysis means to break up, so it's splitting of water using light to form oxygen and hydrogen. Briefly, if we take a look back at the previous slide, you can see that light energy is being used to break up water because the electrons are pulled out of water, and you see oxygen and hydrogen ions are made. So the next assessment statement says, explain the light-independent reactions. Now this is happening inside the stroma of the chloroplast. You see that the carbon dioxide molecules are being combined with a 5-carbon molecule called RUBP. And the name of the enzyme that helps this process is called RUBP carboxylase, or Rubisco for short. This process is a reduction step. And you're making a molecule called which you have to know as G3P. that molecule is converted in a series of steps with ATP and NADPH. They're coming from the light dependent reactions to a molecule that you have to know as triose phosphate. You will have 6 triose phosphates because you had 6 G3Ps. One of those G3Ps is being used to make glucose, which is right there. Of the 6, 5 are being regenerated, right? They're coming back from the dead in a way. Their RUBPs are coming back through ATPs again, which are being produced in the light dependent reactions. And this cycle continues as long as you have CO2 to add to the process. An easy way to remember this is to remember the math. There are 15 carbons here because there's 3 RUBPs and 5 carbon. An RUBP is a 5 carbon molecule. So 3 times 5 is 15. You're adding 3 more carbons here, so that's 18. And you still have 18 at this step because it's 6 G3Ps. And that's 18 carbons total. You still have 18 carbons down here. The molecule's just been rearranged to make triose phosphate. When one of these triose phosphates comes off to make glucose or amino acids, you have 5 triose phosphates left. And that's 15 carbons, which is the same as what you started with in the original RUBP set. The next assessment statement says, state that ATP and hydrogen derived from photolysis of water are used to fix carbon dioxide to make organic molecules. If we go back to our previous figure, what this is saying is that the energy from ATP that came from the light dependent reactions is used to fix the carbon, is used to put it in a form that can be used by organisms. So the next assessment statement says, explain the relationship between the structure of the chloroplast and its functions. We looked at this image earlier when we looked at the structure of a chloroplast, and now that you know where these reactions are happening, we can talk about how the function and the structure are related. So first, there's a double membrane on the outside, and that's containing all the reactions that are happening during photosynthesis. The stroma, which is kind of like the cytoplasm of the chloroplast, allows for the Calvin cycle, the light independent reactions, to happen. The thylakoid lumen is very small, and that allows the H pluses to concentrate inside them very quickly, which means you can create ATP also very quickly. And you see that the surface area of these thylakoids are quite considerable. They're large. And this allows for multiple hydrogen ions to flow across and a lot of photosystems to be present so that ATP can be created. The next assessment statement says, explain photophosphorylation in terms of chemiosmosis. So, there are 2 types of photophosphorylation. There's non-cyclic and cyclic photophosphorylation. The non-cyclic version over here on the left is what you're already familiar with, which I explained earlier. You excite the electrons in both photosystems. The electrons flow through both photosystems, and as you're doing that, Remember, in the middle, when the electrons are flowing through the middle, they're pumping H pluses this way. And then the electrons flow by themselves the other way through ATP synthase and create ATP. The electrons at the end are collected by NADP to make NADPH. When you have enough NADPH in the cell, you don't need to make more. The way that the cell prevents from making more NADPH than it needs by producing more ATP is it uses only photosystem I, and this is called cyclic photophosphorylation. In cyclic photophosphorylation, you are taking the electrons that come off of photosystem I, and instead of putting them in NADPH, you're sending them back to the end of photosystem II, and not including photosystem II, and allowing the H pluses to be continually pumped across the membrane into the lumen of the thylakoid, which when you concentrate the H plus in the lumen of the thylakoid, you allow the H pluses to be pumped across using ATP synthase. Again, I want to make sure that you understand that ATP synthase is not being shown in this image. It's kind of a highly simplified version of it. The last few assessment statements that this video is going to cover has to do with light energy and wavelengths. You can see you have very small wavelengths of light, and you can have very large wavelengths of light. When they're really close together, this is very high energy, and when they're far apart, that's very low energy, low frequency. And the visible part of the light spectrum is actually quite small. You can see that red wavelengths are very low energy, and violet is very high energy. Remember, in physics class you probably learned Roy G. Biv to remember the order of the light in the visible spectrum. It's red, orange, yellow, green, blue, indigo, violet. And the way I remember that violet is the higher energy end is, well, next to violet is ultraviolet. Ultraviolet light is used to kill bacteria in our goggle cabinet. So if that ultraviolet is high energy, so violet is the higher energy end of this visible spectrum, and then red would be the lower energy end. And remember, we use infrared like in our remote controls for our television, so obviously that's very low energy because it doesn't hurt anybody. It says outline the differences in absorption of red, blue, and green light by chlorophyll, and I talk about this in the next slide. You can see here on the left that blue has a very high absorption by chlorophyll. Now, there's different types of chlorophyll. Here shown chlorophyll A and chlorophyll B. And the green wavelengths aren't absorbed much at all. Remember, there's a lot of release of green light because plants are green, but they don't absorb green. They absorb in the blue region. They also absorb in the red region, which is shown right here. So, shining red light and blue light on plants will allow them to absorb light for photosynthesis, but not green light. This assessment statement on this page says, explain the relationship between the action spectrum and the absorption spectrum of photosynthetic pigments in green plants. So each spectrum is a little different in different plants, so that's why this figure looks a little different than the previous one. But in general it's the same. The blue region and the red region have very high absorption, and the green region has very low absorption. Many wavelengths of light are absorbed during photosynthesis, but only some are being used. And the parts that are being used for photosynthesis to take that light energy and convert it into chemical energy, that's called the action spectrum. So you can see on the bottom here is the action spectrum for this particular plant. You see a lot of absorption. You see a lot of light in the violet-blue area that's being used to increase the effectiveness of photosynthesis. it dips in this green to yellow region, and it picks up again in the orange to red region. So the essential takeaway is that in the violet and blue light and orange and red light are very useful in promoting the rate of photosynthesis, where green and yellow light are not very effective in promoting the rate of photosynthesis. Please write down questions, and I will see you in class. Thanks.